Hello dear students, today we are here again with a new video of biology and our today's chapter number 4, Standard 12th, Molecular Basis of Inheritance. Our topic name is Avery, Carty, MacLeods experiment. In previous experiment we have seen about Griffith's experiment. In Griffith's experiment he has used two different kind of strain. One was R strain, one was S strain. R was non-virulent strain and S was virulent strain. And we have seen the experiment on the mouse, that how he has proved the 4 experiments, and in that he has used the different varieties and the combination of R and S strains. Today, here in continuation of that experiment, after the Griffith experiment, Avery, Carty and MacLeod tried to prove that the DNA is the genetic material which is responsible for the transforming principle. What is meaning of transforming principle? Transforming principle means the non-virulent gets the characteristics of virulent. Means R type of strain was being transformed into a S type of virulent strain in the last stage when we have seen in Griffith 4th experiment. So they tried to prove this, that whether a DNA is responsible for this or whether a protein is responsible for this. So he tried the experiment. For this experiment in 1944, after the gap of 10 years of research and experiment, US microbiologist Oswald T. Avery, Colin M. MacLeod and Maclean McCarty, they were all from which university? Rockefeller University. It is in New York. So when they studied after the gap of 10 years or the time duration of 10 years, they came to the conclusion that the first evidence to prove DNA is a genetic material. We know that the DNA is responsible for transforming the genetic character from one generation to the next generation. But it is also responsible for transforming principle of non-virulent to virulent. So he started the theory on transforming principle through his experiments. And how he proved this? They purified the DNA, RNA and proteins from the S strains. S strain was the last strains in Griffith's experiment, where he found that when the mouse was live, that time the R strain was in dominant condition, but when S strain was found, the mouse was dead. But in last case, he saw that the heat killed R strain as well as S strain. When they were in combination, the mouse was alive. So he found that the DNA, RNA and proteins from the S strains, and he mixed it with the heat killed S strain and R strain. Means he has also used the different combination to prove that the DNA is the genetic material. To confirm the transforming principle in R cell and in S cells, what he did, only a DNA is responsible for transforming the S strain, that is the R, to deadly S strain. This word is very important, deadly. Deadly means what? It was virulent. So he tried that R strain was non-virulent but S strain was virulent. So R strain was converted or transformed into which strain? S strain, that is deadly strain. Why? Because of DNA. This word is very important. Means DNA was responsible to transfer. So he tried different experiments and he came to the conclusion of the 3 different kinds of enzyme. The first enzyme was protease. The name itself says protein. Protease. Protease is the protein digesting enzyme. Second is the RNase. RNase is the RNA digesting enzyme. And third is the DNase. DNase is the DNA digesting enzyme. So he tried the 3 different combinations to prove the theory that the DNA is the responsible for the genetic material. In this, DNase was inhibited the transforming principle. Means when he tried the experiment with protease and RNase, he found that the R strain was not converted into the S strains and the mouse was still live. Like here he has conducted the 2 different experiments in case A and in case B. In case A, what he did, the non-virulent RNA was tried with the combination of protease. Protease is the enzyme which digests the protein. So when he tried this combination of non-virulent with protease, he found that the mouse was dead because protease was only digesting the protein, not the DNA. But in second case, he found that when R strain, that is a non-virulent strain, when it was added with a combination of DNase, that is heat killed S strains, which is a smooth virulent one, and S strains was added with the non-strain, that is a non-virulent R strain, he found that the mouse was alive. So it proved that the DNase which was used in the second experiment was responsible for digesting the DNA. That means the virulent or the deadly strain of DNA was digested. That's why the mouse get alive in the second case. And he proved his theory that the DNA is the responsible for the transforming principle. But still his theory was being objected by Hershey and Chase. They were not satisfied. They were the biologists and they tried to prove the next experiment that DNA is not only responsible but also some other factor is responsible. So after the Avery, Carty and MacLeods experiment, the new experiment which came into existence was Hershey and Chase, that we are going to see in my next video. Thank you for watching the video.